Thank you everyone for being here. Thank you very much for that introduction. I was just having a conversation with Alexandra there for a hot minute there, saying that I've been doing this for almost 10 years now. And when I was in school, this mycotoxins in general was not a subject. I never took a mycotoxin course. We never even mentioned it. And really, in the first few years of my career, especially in the poultry industry, it wasn't a topic. So to be seeing multiple mycotoxin symposia during PSA just warms my heart. It's very exciting, and it's something that, as I've been learning more about this field, I've really started to enjoy learning more and more about them. So today, my focus is going to be talking about. I'll touch on a little bit about mitigation strategies, just in general, and what type of options are out there. But my main focus is going to be on the enzymatic technologies. Where did we start? Where are we? Where are we going when it comes to mycotoxin mitigation through the use of enzymes? So I know if you've been to the symposium for the past few talks, you kind of have a basic understanding, if you didn't already, of what mycotoxins are, but they are secondary fungal metabolites. As we've learned more and more about them, we have learned about the existence of more and more mycotoxins. I know this has above 1,000 mycotoxins identified, and I know the number is well over that now. But one of the main issues when it comes to mycotoxins is that they're stubborn, is the best way to say it. They are persistent and they're stable. They're very resistant to chemical treatment, heat treatment. It's very difficult. You can get rid of the fungus, but it's very difficult to get rid of the mycotoxin itself because its chemical structure is very stable. It's been identified in a wide variety of feedstuffs. There hasn't been a feedstuff that I have come across that I haven't found it in. So corn, DDGs, wheat midds, soy oil. You can find it in just about every feed ingredient that you look at. And there are quite a lot of research papers out there identifying the negative consequences of mycotoxins in both humans and animals. And then just the economic impacts to the poultry industry, it's been estimated about $1 billion to the US alone. And that's really just counting mitigation strategies. That's not looking at feed losses. That's not looking at performance and mortality losses when it comes to the birds itself. So really, this is just a drop in the bucket in terms of our estimates of what this is really doing to our industry. So here's just a little image of the major mycotoxigenic molds and mycotoxins. I like to call them the big six, but as we were talking in previous discussions, there are a lot of emerging mycotoxins and even mass mycotoxins that we're just now learning about. So as I said, these are what I call the major six. I like to divide them into two major categories, and it's mostly based on where they are produced. So we have the first major category, what I call field mycotoxins. This is where 95% of mycotoxins are going to be produced, is on the plants in the field before harvest. So your main ones there are going to be your trichothecenes, your fumonisins, and your zearalenone. And then we also have our ergot alkaloids that show up quite a bit in our small grains. And you'll notice, as Dr. Lilleboom was discussing earlier, it's predominantly going to be produced by Fusarium species. And then we have our storage mycotoxins. These are the ones that are going to be produced while that grain is being stored, so after harvest. So we have our aflatoxins and our ochratoxin. And you'll notice that predominantly the fungi that's going to be producing those is going to be your Aspergillus species. So what strategies, what solutions do we have to work with to kind of mitigate this risk? Well, first, as Dr. Glenn was touching on, let's start with the plant. Let's prevent the growth to begin with. Really, that's the ideal scenario, right? We want to start with the plant. We want to prevent fungal growth. So a lot of people look at GMOs as an option, but Dr. Glenn's work that they're doing in their lab and looking at different biological methods to reduce the growth of those fungi, that was very promising, and I'm very excited to see where that research grows. The next step would be decontamination. So this is either through a physical or a chemical treatment. But as you can imagine, this is going to be very time consuming, very labor intensive. And just because these chemical structures are so stable, it's really not an effective means. Dilution, I'm sure you've heard this phrase many times. I'm sure we could all recite it. Dilution is the solution, right? And in many cases, that is true. But what does that entail? That's going to entail a lot of labor. We're going to have the facilities to be able to take different amounts or different piles of corn, for example, and to be able to mix them. That takes additional facilities. But you also need to know what you're working with. How do I know that this is a low-level corn that I want to mix in? How do I know this is low-level corn that I need to dilute down? Sorry, there's a lot of yelling going on behind me. So that also requires testing and making sure you get a representative sample to ensure that the results that you are getting are giving you a adequate assessment of the risk that you're dealing with. So really that leaves us with what currently the majority of our industry is using, and then that's mitigation through the use of feed additives. So this is using a feed additive that's going to go into the animal that's going to help reduce either break down those mycotoxins, either break them down, bind them so that they're excreted, or biotransform them so they're in a non-toxic form, or help just with bioprotection, helping that animal get over that challenge a little bit more by, say, protecting the immune system or protecting the liver, which tend to be the main systems that are going to be affected by mycotoxin challenge. So here's just a brief overview of the types of mitigation products that you can find out there. I will say most of the time when you talk about mitigation feed additives, the number one word I hear is binders, right? Binders. So this would be, I think, the largest category out there. The most predominantly used category is binders. And there's a couple different options which one's the laser pointer? This one, there we go. Oh, that's not what I wanted. There we go. There's a couple different categories within that binder category. You have your inorganic binders. Those would typically be like your clay minerals, your aluminosilicates, your bentonites. Then you have your organoclays, such as organozoolites. Then you have your organic binders. This would generally be your yeast products, your yeast cell walls, your beta-glucans, your moss products, for example. Activated charcoal, if you've ever had a pet eat something that it wasn't supposed to and had to go and give your animal activated charcoal. Obviously, that's not something that we use in the poultry industry, but it is an option. Next would be your chemical options. The main one that would be on the market is sodium metabisulfate. As far as I'm aware, we do not use that in poultry. It is only used in the swine industry, but it would fit into our feed mitigation product category. And the last is your deactivators. So these are either microbes or they're enzymes that are going to physically break down a mycotoxin into non-toxic metabolites. And I'll touch on those here in a little bit. So here's a study that was done quite a few years ago. It looks like it was published in 2015, where we looked at a number of different mycotoxin mitigation products and looked at their ability to bind aflatoxin in this case. You can see we've got aflatoxin on the x-axis, or the percentage of adsorption or binding, and at different pHs, pH of 3, pH of 6.5, to kind of look at different pHs that you may find throughout the gastrointestinal tract. So here, the first group, let me bring these up. This group of products would be considered inorganic binders. So again, these are your clays, your bentonites, your aluminosilicates. These were the organoclays, and these were the organic binders. You can see that for the most part, the inorganic clays and the organoclays work great when it comes to aflatoxin. The organic binder is a little bit more variable. It just depends on which fraction of the yeast cell wall, the yeast in particular, it tends to be a little bit more product specific. We did notice that there was a group that had a higher amount of adsorption ability. And if we did a further chemical analysis on those, we did find that those had a high ash content, which meant that they likely did have a clay component in them that was probably likely doing the binding itself. Now, the same assessment was done with the same group of products, but for deoxynivalenol. You can see, I don't even have to point out which ones were which, it did not matter. You can see a very stark difference. So why the difference? Why the difference? And it really has to go down to the chemical structure of those two different mycotoxins. So let me look at, these are the chemical structures. Let me pull up aflatoxin here. If we look at aflatoxin is planar. It's flat. It's two-dimensional, if you will, and it's polar. So if you look at a clay, for example, you look at it under a microscope, you will see that there are multiple sheets kind of stacked on top of each other. And so because it is planar, aflatoxin can fit within the sheets of that clay. Because of its polarity, it's bound with high affinity. Then it is pretty much permanently attached, and then it's going to be excreted out with the feces, so then the bird is not going to be absorbing it in the intestine. Now, what about Dawn? Dawn was the one that, regardless of the type of binder we were using, we really didn't see any adsorption going on. And it's because it's not planar. It's three-dimensional, if you will. It's very bulky. So it's not planar. It does have polarity. So in some cases, you would be able to bind it depending on the structure of the clay. But because of its three-dimensional structure, it's not able to be bound by a typical binder. And the analogy I like to use is imagine a parking deck. That's your bentonite sheets. Your aflatoxin is your nice little commuter car, fits nicely in that parking deck. Deoxynivalenol is your Mack truck. It's not going in there no matter how hard you try and get into that parking space, get into that parking garage. It's not fitting. So that leaves us with, well, what do we do about deoxynivalenol, for example, or those other mycotoxins that cannot be bound by a typical binder? So not all toxins are the same, which means your strategies also need to change based on the challenges that you're facing. And this is just a schematic to kind of show you. We've done quite a few different adsorption analyses to kind of figure out which ones were more adsorbable than others. Aflatoxins obviously being your most adsorbable, deoxynivalenol being your least adsorbable, and then you have some of the others down in the middle, fumonisin, zearalenone T2 being some of the ones that are not as adsorbable. So what are the other options? And this is where we're going to get into enzymatic technologies. And the use of enzymatic technologies has been given the term biotransformation. So it's the deactivation of functional groups, so those groups that give the mycotoxin its toxic traits, of the mycotoxin into non-toxic metabolites, thus rendering it non-toxic. So here's just a little example. You've got your mycotoxin, you have your enzyme coming in, breaking, cleaving it down into non-toxic metabolites. Now, why would we want to use enzymatic technologies? Well, one, it's specific. We can target specific parts of the chemical structure to break it down. It's irreversible. We don't have to worry about the mycotoxin coming back together and becoming toxic again. And it's going to be very efficacious against those mycotoxins that are non-adsorbable. So the concept of biotransformation is not new at all. This was actually one of the original papers. I believe it was Sigler et al. And you notice the date on it. I highlighted it up at the top, 1966. And if you remember some of the papers that Dr. Lilburn showed, you notice it was just around the same time in the early 60s when aflatoxin was really first identified with turkey X disease. So they did a analysis of 1,000 different microorganisms, including yeast, molds, bacteria, algae, so on and so forth. And they found one bacteria that was able to completely detoxify aflatoxin in milk. So even back in the 60s, we were looking at microbial biotransformation as a mitigation strategy for mycotoxins. So that was back in the 60s. It's kind of become a little bit more commercial, if you will, in actually producing products consistently that could be used on the market for mycotoxin mitigation. So here I have an example of a microbe. In this case, it's called Biomin BBSH797, which was a microbe that was chosen because of its ability to break down trichothecenes. So this particular bacteria, I'll give you a little fun fact, a little history fact. BBSH 797, you can see it down at the bottom, is actually named after the scientists that characterized it. So it stands for Binder, Schatzmeier, and Heidler. It's actually two couples that happened to, I don't know how they worked for so long together, two couples working in the same lab. Three of the scientists are still actually working with us at DSM-Firmenich, and they characterized it in July of 1997. So that's where that name came from. But this particular bacteria is an anaerobic bacteria that was isolated from the rumen. And it was chosen because of its ability to efficiently produce deoxydases. So if you look at the structure of trichothecenes, every one of them, regardless of whether it's deoxynivalenol or T2, they all have this epoxide ring structure right here. So this bacteria produces an epoxidase that's going to break that ring and create a detoxified form. So this structure is actually deoxynivalenol. We're going to break that ring, and we're actually creating a non-toxic metabolite called DOM1. So what's the next step forward? We have bacteria that are creating these enzymes. Well, why not just go straight to the enzymes? Can we do something that's even more efficient, even more tailor-made? So the next step is looking at fumonisn degradation. If you remember from some of the talks this morning, I believe it was in Alexandra's talk, fumonisns is one of the most prevalent mycotoxins in the world. In the US survey, I believe we're consistently over 80% of our corn samples come back positive for fumonisn. So that was the next target for our scientists, and looking at a enzyme technology that would help break down fumonisn. So in order to create an enzyme to break down fumonisn, you must first know what you're dealing with. You need to know how that mycotoxin works, what is its mode of action, and are there any biomarkers that we could use to assess the efficacy of a mitigation strategy. So here, this is a basic schematic of the mode of action of fumonisns. So fumonisns alter sphingolipid metabolism. So sphingolipids are very important in surrounding nerve cells. They're also components of muscle and nerve cell membranes, so they're very, very important in the body. So fumonisns come in. They are going to block ceramide synthase. It's an enzyme that helps break down free sphingonines and sphingosines into other complex sphingolipids. So if we break down, or if we prevent the production of ceramide synthase, we're going to have an increase in sphingosine and free sphingonine. It's actually at a disproportionate ratio. So we have more sphingonine show up in the blood than sphingosine. And that ratio, what we call the SA-SO ratio, we actually use as a biomarker to look at the effects of fumonisn and potential mitigation strategies for fumonisn. Oh, and thus we have less complex sphingolipids. Okay, so how do we create this enzymatic solution? What is the pathway? So first we have to find, as we touched on before, we have to find a microbe. We have to find something that already produces that enzyme that we want to use. So we're going to look at, usually these microbes are going to come from the environment, usually soil. We find a strain that's very efficient at producing the enzyme that we're interested in, do a genomic analysis of that microbe, and look for those genes that are responsible for producing the enzyme that we're interested in. Then we're going to express that enzyme into a carrier host. It could be E. coli, for example. Might need to do some enzyme engineering, but then we're going to ferment them in once we get the enzymatic engineering down, then we go into fermentation, then we go into the testing process. What does this do in vitro? What does this do in vivo? And then we ultimately go to a product formulation that could be used on a commercial level. So this is going to be, I'm going to kind of tell you the story of our fumonisn enzyme that we have. First, as I said, we're going to identify that strain. So by looking at hundreds of different microbes, we identified a strain of Sphingosis, MTA-144 to be specific, that was isolated from composted earth, that was a very efficient producer of fumonisn carboxylase. And you can see here, this was a in vitro analysis looking at the ability of this particular strain to break down fumonisin. So here, the black bar is fumonisin. The red bar is hydrolyzed fumonisin B1, which is the byproduct of enzymatic breakdown of fumonisin. So you can see just within a few hours of being in contact with the strain, the amount of fumonisin drops pretty dramatically, while the byproduct, if you will, increases quite quickly. So we're seeing 100% breakdown of fumonisin with that particular strain. So the next step is taking a closer look at the genome of that strain. And after doing that, they identified that the gene cluster responsible for fumonisin degradation was identified as, it's called FUMD, F-U-M-D, which encodes for the carboxylesterase that breaks down the fumonisin. So we take that and then we put it into this host. I'm not going to try and pronounce it. And we produce that enzyme. So that enzyme has been coined FUMzyme. FUMzyme was EFSA approved quite a few years ago, I believe early, like in 2015 or so by EFSA. It was approved by FDA in 2021, 2022? Thank you, Paige. 2022 for swine and poultry. So this was the first ever FDA approved mycotoxin mitigation ingredient. And as I said, it was for swine and poultry. It is a fumonisin esterase. It is a purified enzyme that breaks it down into fumonisin. It breaks those TCA side chains that you see here. So we have two TCA side chains, and that hydrolyzed fumonisin B1 backbone is left. So the next step is in vitro. What can we see in vitro? Here we have the orange bar is your fumonisin. Your green bar is that hydrolyzed fumonisin B1 backbone. The black bar is your TCA side chains. So here they took 72 ppm of fumonisin B1, which I've seen very high levels of fumonisin B1. I don't think I've ever seen 72, so this is a very, very high level. And exposed it to 17 units of FUMzyme. And you can see that just within 15 minutes, it was able to completely break down that large amount of fumonisin into its non-toxic metabolites. And then finally, what is it doing in the animal? So here, this is a broiler study. They used Ross 308s. We had a control group that was fed a standard basal diet, a group that was given a contaminated diet. It was contaminated with 15 ppm of fumonisin, and then a group that was given 15 ppm of fumonisin plus one, we would consider one dosage of 15 units of FUMzyme. And we looked at the amount of fumonisin being excreted in the feces of those birds. And you can see here the amount of fumonisin in the feces of those birds significantly increased. This is only after 14 days of exposure. Increased in those challenged birds, but then significantly decreased when FUMzyme was brought in. And then here, if we look at hydrolyzed fumonisin B1, so this is the metabolite, we don't see it in those first two groups as we would expect, but we do see it increase in that FUMzyme treated group. So what's next? So we went from microbes producing enzymes to recombinant enzymes for fumonisin. What's next in the pipeline? Where are we going? And next would actually be zearalenone. Zearalenone, I would say, is number 3 in terms of our mycotoxin challenges out there. And we touched on them a little bit in the previous talks about how this can dramatically affect reproductive animals. So that's going to be the next one that's in the pipeline. So I'll touch on a little bit of that process. I don't want to get too much in the details because it's a little bit, it's very similar to the FUMzyme process. But the research that's been going on just in terms of looking for microbial strains that degrade Zen has been going on for quite a few years now. So these are a few papers that were published looking at the ability of different strains of Gliocladium roseum that is able to break down zearalenone. So this is 3 different papers, and it also showed the metabolism of zearalenone by these enzymes. So you can see we go from zearalenone to hydrolyzed zearalenone to decarboxylated hydrolyzed zearalenone. There we go. So we've kind of identified some microbes. There's obviously more out there. But as they were looking at these microbes, they again looked at the genetic material to identify what was actually giving these microbes these characteristics. And they identified that it was producing a lactohydrolase. And the gene that was responsible for producing that enzyme was called ZHD101. There we go. So we already touched on this before, the process, but it's a very similar process, just a little bit different strains that we're using as the one that we got the genes from, or the genetic material from, and the ones that we are using to express, but I'll get a little bit further in here. So starting with the in vitro analysis of that lactonohydrolase, the ZHD101, that's the gene I told you that was calling from the previous microbe that we know helped produce that enzyme from those two previous papers. But they were able to find another enzyme. I thought I had a little piece that said what it was called. I think it's called Zen-A, that they actually found was much more efficient and had much greater affinity for zearalenone. So here, this was the original gene that they found, or the original enzyme that they found. This is the catalytic rate of breakdown for zearalenone, and I would pay most attention to that x-axis and the units there. So this is the same data. This is that blue bar. This blue bar is this blue bar. This is the Zen-A, also known as Zenzyme, that was isolated from a strain called PFA-D8-1. So you can see much higher affinity, much faster catalytic rate. And then looking in vivo, what is this? How is this enzyme working in the bird, and how is it breaking down in the different organs? So here, this is looking at the crop and the gizzard. They removed the digesta from these birds. The birds were given different increasing amounts of Zenzyme. And you can see here, just looking at the amount of zearalenone in the crop of those birds, it's going down in a linear fashion. But then if you look at the breakdown components, the hydrolyzed Zen and the DH-Zen, we can see that that's increasing as we would hope it would. And we're seeing a similar effect in the gizzard, where zearalenone is decreasing and the other two metabolites are increasing. This product has been used in a few countries now. I believe it's been used in Asia and maybe some parts of LATAM now. Hopefully it'll come to the US eventually, but not as of yet. So that kind of brings me to the end of my presentation. If we have time, I'd be happy to take any questions. If not, I will be around, and feel free to approach me. If you ever want to contact me, my email address is up there. Feel free to reach out at any time.